Yes, sir.
And today, we are here to discuss the cell surface and extracellular matrix.
Osmosis is a form of passive transport when water molecules move from low solute concentration, high water concentration, to high solute for low water concentration. A cross membrane is not permeable to the solute. There is a form of passive transport called facilitated diffusion. Sa atong nakita karon natin baso, and then atong ibot ng asin. This is a kabalin sa pikas kay naman sa wall. Pangan kay semi-permeable membrane.
So imbis ang asin, ang sodium, pang molecules mo balhin, ang tubig ni balhin. So atong nakita danhi, bisig masaghan pa ni siya kuan asin, nimubang tubig pareha na sila kasikip. So balance na.
So, mo din yung transmembrane protein kaning orange mo ni ang responsible sa osmosis.
O so ni ang kaning transporters kita mo ani manay tig suyop sa kuan sa mga molecules. Kunya, di ba passive man atong gamit nga kuan kanang osmosis. Mao ni anhira siya mo agi sa mga kuan, lusot na siya ba. Kaya dili man siya kuan mo lusot ang tubig depende asa ang high level of concentration. Kung nasud, ang tubig mo sud. Kung nasgawas, ang tubig mo gawas.
Endocytosis is a cellular process in which substances are brought into the cell. The material to be internalized is surrounded by an area of cell membrane, which then buds up inside the cell to form a vesicle containing the ingested material. Endocytosis includes pinocytosis and phagocytosis. Ang endocytosis di ay sida ang magda sa mga material padong sa cell o mo-absorb. Mo rin na sida.
Presong nakita sa kaning maong diagram. Kung kabantay mo, let's say, kung ani ka ng potassium, mao ni siya ang cell wall. Kung mao siya kitawag bulk, kay kung kabantay mo, daghan gud siyang murag nasud, kay pareho na sa shipping, bulk shipping, daghan na siya gipang ship. Like sa giingon ani, kanang the material to be internalized, kani silang mga kuan material or mauna, ma-surrounded siya kuan area of cell membrane. Kadugayan, murag masud jud siya, mahimo siya physical niya. Kuan niya i-absorb jud niya.
Exocytosis. Exocytosis is a process for moving large molecules out of the cell to the cell exterior. Commonly, these macromolecules originate in storage vacuoles inside the cell and are moved to the exterior after an appropriate signals for this action. So, ang exocytosis is binalira ni siya sa endocytosis, which is endo is pasod
In
and exo is pagawas.
exit. Pareha sa naa sa diagram. Uni ang storage vacuole, maning cell membrane, kadugayan, mugawot din siya.
Cell adhesion. Cell adhesion is the process by which cells interact and attach to neighboring cells through specialized molecules of the cell surface. Which is, cell adhesion is the binding of a cell to another cell or a cell to an extracellular matrix component.
So, what is ECM? The extracellular matrix, or ECM, is an intricate network composed of an array of multi-domain macromolecules. Organized in a cell or tissue, specific components of the ECM link together to form a structurally stable composite, contributing to the mechanical properties of the tissues. So, ECM is a complex structural entity surrounding and supporting cells that are found within mammalian tissues. So, basically, kasagaran, ang ECM makita sa mammals like us. So, ang ECM, usa ka pundasyon sa cell. So, kay pundasyon man siya, ECM is to provide and support. So, unsa man niyang i-provide? ECM provide structural and biochemical support. So, there are three roles of ECM. Scaffold, formation and maintenance, changes cell behavior in response to environment, and use in cell-to-cell communication.
The function of the extracellular matrix or ECM. Function. Due to its diverse nature and composition, the ECM can serve many functions, such as providing support, segregating tissues from one another, and regulating intercellular communication. The extracellular matrix regulates a cell's dynamic behavior. So as stated before, yung uses kay scaffolding, which is structure, communication, and maintenance. Essentially, ang ECM is like spinal cord nato. Siya yung murag give orders. Kaya ang brain man mo ka na to, no? Ang muhatag o example, palihupon yung kamot. Muagi siya sa atong kuan, sa atong spine, o atong nervous system. Siya yung murag mo command unsay buhaton sa mga cell. Example, akong mga classmate ang hikay mga cell. Ang ECM, siya tingnan nga, oy, ang hika, maning buhaton, oy, anha ka, maning buhaton, o siya tigkuan ka ng tig-repair, ana. So, gamit yung kaning ECM niya, intricate case siya, daghan kasi mga components. As stated before, it also serves as a determining and controlling factor in fundamental behavior and characteristics of cells, such as adhesion, migration, polarity, differentiation, and apoptosis. As stated before, as in grabe kayo siya kagamit, guys. Siya bahala sa adhesion, as nakadiscuss natin before, migration, muagi, polarity, and so on. Parts of the extracellular matrix and its function. Ako nang mga classmate ni guys.
On the other hand, parts of the extracellular matrix and its function. The first one is the fibronectin. Fibronectin is a high molecular weight glycoprotein of the extracellular matrix that binds to membrane spanning receptor proteins called integrin. It is also a multifunctional adhesive glycoprotein that plays an important role in tissue repair, in regulating cell attachment and motility, and in embryogenesis.
So guys, fibronectin is, of course, as stated, it binds to the integrin, which connects the cell's cytoskeleton to the ECM, and it is also, yes, again, as stated, important in regulating repairs by adhesion and all that. Kundi guys, ang ECM guys, it's not just make up sa ang nahimo nga ang written down nga mga molecules. Daghan kini siya guys, pero di na mo i-add yung mga redundant. Kay example, collagen 4, capital collagen 3, redundant na guys siya guys ba? So what ano yung exclude mo? Yung main mga components. Next slide.
Laminin. Laminin are a family of glycoproteins of the extracellular matrix of all animals. They are major constituents of the basement membrane, namely the basal lamina. Laminins are vital to biological activity, influencing cell differentiation, migration, and adhesion.
Back to the slide, please. As we can see guys, ang laminin kay na ay alpha, beta, and gamma parts. I will further elaborate on that later on the video guys.
Collagen, which is constitute the main structural elements of the ECM, provides tensile strength, regulates cells adhesion, supports chemotaxis and migration, and direct tissues development. Collagen is a fibrous protein. Collagen is found in our skin, hair cells, connective tissues, and bones, and etc. Collagen is the major protein in the extracellular matrix. Collagen is a large family of proteins having different subtypes. So there are two subtypes of collagen. One is fibrillar and non-fibrillar. Fibrillar collagen, which includes type 1, type 2, type 3, and type 9. Non-fibrillar, which has associated collagens, short chains, and basement membranes. multiplexing which has multiple helix domains and membranes associated collagens. So, there are 5 types of collagens. Type 1, found in skin, tendons, and vasculate organs, bones. Type 2, found in cartilage. And type 3, found in connective tissues. And type 4, found in basal lamina. Type 5, found in a cell surface and hair and placenta. Collagen is a protein molecule made up of amino acids. It provides structural supports to the extracellular space of connective tissues. Due to its rigidity and resistance to stretching, it is the perfect matrix of skins.
Next is collagen 4. Collagen 4 is a type of collagen found primarily in the basal lamina. The collagen 4 C4 domain at the C terminus is not removed in post-translational processing, and the fiber link head-to-head rather than in parallel. So, collagen 4 is a type of collagen that plays a key role in forming the structural framework of basement membranes. Especially in the basal lamina is a specialized extracellular matrix that provides support to various tissues. Next is tenascin. Tenascins are classified as modulators of cell adhesion, migration, and growth. Type 3 modules in tenascin as well as fibronectin bind to VEGF and stimulate cell formulation in an integrin-dependent manner. So, tenascins are protein know their involvement on regulating various cellular activities such as adhesion, migration, and growth. Within tenascins, there are type 3 modules which are circular component.
The next part is decorin. Decorin is involved in an extensive signaling network and can interact with other extracellular matrix components, growth factors, receptors, tyrosine kinases, and various proteases. Decorin has been shown to be involved in wound repair, cell cycle and angiogenesis, tumor metastasis, and autophagy.
So, ang decorin ay sila day ang kuan. Important ka do kay sila ang mag-repair sa mga day ug musamad ka gamit sa kay sila ang mo-repair og kuan.
So the next part is the glycoprotein. Next slide. So glycoprotein perform vital biochemical and structural functions. They enable cells to recognize another cell as familiar or foreign, which is called cell-to-cell recognition. They also help cells attach to and bind other cells, which is called the cell adhesion. Glycoproteins of the extracellular matrix and basement membrane play a crucial role in cell recognition, adhesion, migration, and proliferation. So, the glycoprotein are proteins which contains oligosaccharide. Oligosaccharide chains covalently attached to amino acid chains. So, there are carbohydrates is attached to the protein in post-translational or post-translational modification. So, this process are known glycosylation. So, ang glycoprotein essential siya sa reproduction, and there are many examples of the glycoprotein, which is the collagen, the mucins, transferrin, ceruloplasm, and etc. That's all the glycoprotein.
So the next part is osteopontin. Secreted OPN binds to integrins and CD44 as well as to several extracellular matrix molecules including fibronectin and collagen. OPN has also been shown to regulate the extravasation, phenotype, activation, and survival of multiple immune cell types.
Next is the protoglycans. Protoglycans that are heavily glycosylated, the basic protoglycan unit consists of a core protein with one or more covalently attached glycosaminoglycan component. So, the function of the protoglycans help protect cells from injury by outside forces. Next is the integrin. Next slide, please. So, the integrins are the principal receptors used by animal cells to bind to the extracellular matrix. They are heterodimers and function as transmembrane linkers between the extracellular matrix and the actin cytoskeleton. A cell can regulate the adhesive activity of its integrins from within. So, integrins join the cytoskeleton on the inside of the cell. So, ang cytoskeleton, nag-connect siya sa inside the cell. And the outside is the extracellular matrix. So, maano cytoskeleton. So, nakakonect siya, which is the integrins. Sa gawas, kakonect siya, which is the extracellular matrix.
Next is aggrecan. Aggrecan is a protein found in cartilage, like joints, knees, backbone. Aggrecan is a major protoglycan in the articular cartilage. This molecule is important in the proper function of the articular cartilage because it provides a hydrated gel structure. It's the cartilage with load-bearing properties. Aggregans provide intervertebral disc and cartilage with the ability to resist compressive loads. Next is fibrous protein. Fibrous protein is usually static molecules that plays important structural roles in anatomy and physiology or vertebrates, providing external protection, supports shape and form. They are water insoluble and are typically built upon a single repetitive structures assembled into cables or threads.
Now we move on to the structure of the extracellular matrix. Next slide. Over here, as we discussed, we can see the integrin, and we know the function of the integrin is to connect the cytoskeleton of the cell, inside the cell, to what's outside the ECM, the extracellular matrix. Next slide. There we go, the ECM and the cytoskeleton. Next slide. Laminin belongs to the family of the glycoprotein, and they are an integral part of the structural scaffolding in almost every tissue of an organism. You may ask, where can we get laminin? But they are actually secreted and incorporated into cell-associated extracellular matrices by cells. Laminin is vital for the maintenance and survival of tissues. Defective laminins can cause muscle to form improperly and lead to muscular hypertrophy. So, as you can see, this is the laminin. We have the alpha, beta, and gamma. They actually have their own functions. The alpha is actually responsible for interacting with integrins, and the beta and gamma are responsible for self-assembly. The bottom part, the coil part, can also interact with integrins, perlicans, and dystroglycans. All of this combined can create an intricate structure. As we can see here, it's like a web. Next slide. How laminin and collagen are interconnected to each other. So we can see the laminin, all that. The other parts of the ECM that we haven't mentioned in this structure are actually they're not as important because they act as like cement. They're just there to solidify the bonds. These are the actual structures. Like in a building, we have the pillars. Next slide. How laminin and collagens are, there we go. With these intricate connections with laminin, nitrogen, and collagen, we have this structure. Next slide. And with that, all combined, we have the ECM. To the integrins, laminins, and all that, we have the ECM. Everything is connected.